Good morning. In the last part of the last video, we have discussed quorum sensing or talk between bacteria, and we explained clearly that this communication between bacteria occurs through chemical molecules that bind with receptors, and then the result of that talk was the activation or deactivation of certain genes. What we have discussed on the previous slide is an example of cell signaling, or the third type of cell-to-cell communication. And cell signaling might be local signaling in the vicinity of the cell, in the nearby environment of the cell, or it might be remote signaling, meaning that it could signal molecules to distant areas of the body away from the cell. We will start by local signaling, and there are several benefits of local signaling. Let's see first how each type of local signaling occurs and how this might benefit the cell. The first example of local signaling is the signaling that occurs from one cell to itself. So it is the release of local signaling molecules. And we have agreed that the signaling molecules are molecules that, if you're ever asked about the meaning of a signaling molecule is a molecule that can bind to a receptor. And that receptor might be inside the cell, in the cytosol of the cell, or it might be on the cell membrane of the same cell, or on the cell membrane of another cell. And this signaling molecule will arise from one cell and will stimulate or inhibit the same cell. This means that the signaling molecule will bind to receptors that are present on the same cell. In such a case, this local signaling will be known as autocrine signaling. Now, auto means self, meaning that the cell is sending signaling molecules to itself, but it might also be to other different cells. So in such a case, if the molecule, the signaling molecule, is sent to a nearby cell, this is called a paracrine cell. And meanwhile, it is called local signaling because this cell is a neighboring cell that lies in the nearby neighborhood of the producing cell, or the cell that produced the signaling molecules. When these signaling molecules bind to receptors, and they have to do, because if the signaling molecule does not bind, suppose this is a receptor, and this is the complementary signaling molecule. They should be complementary in shape, and they should be capable of making weak bonds with each other. So if this signaling molecule, that we can simply call a signal, does not bind with the receptor, does not fit in, then it will not exert its effects. When it exerts its effects, this might be autocrine or paracrine, meaning that it might be inducing changes in itself, in the producing cell. For example, the cell might be activated or might be deactivated, as we have seen in quorum sensing. The bacteria might be activated and might activate the luciferase enzyme to produce luciferin, and therefore bioluminescence can occur. Luciferin is a protein. Luciferase is another protein, but luciferase is the enzyme, and luciferin is the product, as we discussed in the previous slide. or it might induce other cells to grow in size, to divide, to contract if they are muscle cells, to release a secretion. And this is what we call paracrine stimulation. But it might also activate or deactivate itself, just like it can activate or deactivate other cells as well. Those are the important functions of signaling molecules, or the important effects of signaling molecules and the domain where they act. Examples include growth factors. When growth factors, which are considered signaling molecules, so growth factors are the first example of signaling molecules. They are produced by one cell and induce a nearby cell to divide, and first to grow in size and then to divide. So this is one example, the growth factors. And notice that the growth factors are not the growth hormone. The growth hormone is a hormone that is secreted by the anterior lobe of the pituitary gland into the blood. Pituitary gland is the master endocrine gland in the body. But the growth factors can be produced everywhere in the body, from many cells in the body. So the growth factors are molecules, signaling molecules that bind with receptors of nearby cells to induce them to grow and divide. Okay, and then we have the neurotransmitters, and the neurotransmitter hormones are peptide hormones that are released from the terminal branches of one nerve cell, of one neuron, to stimulate or inhibit an adjacent neuron, or to stimulate an adjacent muscle, or to stimulate an adjacent gland to release a secretion. Because when you study the nervous system, you discover that the neurons, that the motor neurons have an axon, which is a protoplasmic projection, and it has terminal branches. And of course, the cell body here has minute, small, highly branched processes. These processes at the end are, this is the axon, and this is the cell body. The terminal arborization, or the terminal branches of the neuron, have swellings, and these swellings have vesicles, and these vesicles secrete chemical substances which are called neurotransmitters. Neuro because they are secreted by a neuron, and transmitters because they transmit the effect from one neuron to another. So these neurotransmitters may stimulate another adjacent neuron, or they might even stimulate a nearby muscle, or they might even stimulate a gland. All these three structures, the neurons, the muscles, and the glands, are considered, especially the muscles and the glands, are considered effector organs because they achieve or implement the orders that are given to them by the motor neuron. This space that lies between one neuron and another is called the synapse. So the neurotransmitter molecules, whatever their type is, are released by a presynaptic neuron, a neuron that comes before the synapse, and they stimulate a postsynaptic neuron, or a neuron that comes after the synapse. Just like we have autocrine and paracrine cell signaling, the signaling molecules may flow or be transported to large distances across the body, and this is what we call long distance signaling or remote signaling. Why remote signaling? Because the affected organ, the organ that will be affected by the signaling molecules, lies far away from the producing cells. And the first interesting example is at the growing tip of the stem of a plant. If this is a young stem that is still growing up, at the end of the stem, we have active cells that are called active meristematic cells. And as we know, we have previously explained, the meristematic cells can divide into any other type of cells, and they are rapidly dividing. So these active meristematic cells divide rapidly in order to ensure the growth of the stem. But meanwhile, the tip of the stem contains cells that have that can secrete signaling molecules. And these signaling molecules are called the auxins. And these signaling molecules are not released from the plant, but they diffuse from the producing cell to other areas of the plant, for example, to the leaves, to the flowers, etc. And these auxins are considered an example of remote signaling. They don't only control the growth of different tissues of the plant, but they also control the movement of plants. For example, you can see that the stem like bends towards light if the stem receives light from one direction. So this effect on the stem that is affected by light is called tropism or phototropism, and it is one of the effects of auxins on the plant. It is one of the effects of auxins on the plant. This movement of the stem of the plant towards light is what we call positive phototropism. Positive phototropism, and auxins are responsible for this positive phototropism of the plant, and it helps, of course, the plant to benefit the maximum from the light intensity to which it is exposed. In animals, we have an example, a famous example of remote cell signaling. I've just explained the action of the pituitary gland as an endocrine gland whose cells secrete hormones, and these hormones are released into the blood. So we can say that growth hormone is an endocrine secretion, of course, but it is a remote signaling molecules. But growth factors are not remote signaling molecules. They are local signaling molecules, and this is an important difference between growth factors and the growth hormone. Also, when you study the immune system, you will find that we have two types of immune cells. We have the B cells that secrete antibodies against the microbes or the pathogens, and we have T cells. And T cells usually attack the microbe themselves without secreting antibodies. But there is a type of T cells that is called the helper T cell, and that is actually a helper for both B cells and T cells. And the main function of this helper T cell is to alarm the whole immune system that the body has been invaded by pathogens, and it secretes chemicals that travel to distant areas to stimulate the B cells and T cells and awake them and tell them that the body is exposed to danger. And now we are going to talk about a very important concept, which is the signal transduction pathway. Transduction means that you're going to convert the binding of the signaling molecule to the receptor on the cell membrane or inside the cell membrane to a cellular response that is amplified and that is nothing but chemical reactions. So if I have a cell like this, and let's start by receptors that are present on the cell membrane. So this signaling molecule, suppose this signaling molecule is a growth factor, for example. The growth factor is a peptide, so it is hydrophilic, and it will not be able to pass easily across the cell membrane. So it will have to manage to induce a response, a cellular response inside the cell without itself, the growth factor, being forced to enter the cell, because it will not be capable of entering or passing across the cell membrane on its own. So what happens is that this signaling molecule, which is the growth factor, will bind with a receptor on the cell membrane. And this is the first stage, that is reception. And when reception occurs, there will be changes in the receptor. They will be changes in shape or changes in the conformation of the receptor. And this change in the shape of the receptor will induce a cascade of chemical reactions or molecular interactions. And this is actually where we have transduction. This is actually the main body of the signal transduction pathway, which is the step called transduction, in which you translate this binding between the growth factor and the receptor into a series of molecular reactions. And when you have a series of molecular reactions, you finally obtain a response. And that response, that cellular response, is usually a change that is made in the cell. It might be a change in the shape of the cell. It might be for example, storage of material to growth. It might be inducing the cells to carry out secretion. It might be different things. It might be an increase in the rate of metabolic reactions, or it might be a diminution. This is the cell response. And usually the cell response is magnified, meaning that if you start by one molecule, for example, and you are supposed to obtain two molecules as a product of the first reaction, at the end of this pathway, which is actually a cascade of chemical reactions, you will have thousands or millions of molecules. So there is this important concept of amplification, and it all depends upon a series of chemical reactions. But a cascade or a chemical pathway is a series of chemical reactions, but they are interrelated chemical reactions, meaning that reaction A will produce products that will be the reactants of reaction B. Reaction B will produce products that will be the reactants or the reagents of C, and so on. And every time, the number of molecules obtained is amplified or multiplied. So whether you're talking about a local signaling molecule or a remote signaling molecule, they both can activate a signal transduction pathway. So this applies to both local and long distance cell signaling molecule. The signal transduction pathway, as we said, is a complex, of course, process. Because why is it complex? Because it involves many chemical reactions, and it is a multi-step process. And it consists of small number, we start by a small number of extracellular signaling molecules. Why extracellular? Because they are hydrophilic, so they cannot penetrate the cell membrane themselves, and these signaling molecules will produce at the end of the day a major cellular response. And this is why we call it a cascade effect. A cascade effect does not only mean that reaction A will stimulate B or B will stimulate C, but that there will be an amplification of the response. Like in dominoes, when the first piece of domino falls, it results in the successive fall of the other pieces of dominoes. So at the end of the day, all of them will be moved or fall down. Now, the signal transduction pathways, therefore, result in an amplified cellular response. So you can start by one signaling molecule and you end up by having thousands of molecules, and these are the product molecules. Now, the ligand here, the term ligand refers to the signaling molecule that binds to the membrane receptor. If the signaling molecule, as we said before, is hydrophilic, so the signal transduction pathway should involve 3 stages, because the first stage, which is the stage of reception, will be a necessary stage, because a signaling molecule that is hydrophilic is not capable of passing through the interior of the cell membrane, which is hydrophobic. So how would it work out this issue? By binding with a specific receptor on the cell surface, and therefore the specific receptor, as we have just explained, will undergo a change in conformation. And after having made this change in conformation, there is the transduction, meaning that we will change the binding of the signaling molecule to the receptor, which is the signal, into a cellular response. How? By inducing a cascade or a series of molecular interactions. That will start by what? That will start by change in the conformation of the membrane protein. So this change in shape of the receptor, so the first step after binding is that the receptor changes shape. And when the receptor changes shape, this will activate a famous enzyme called adenylate cyclase enzyme. As we said before, adenylate cyclase enzyme. And adenylate cyclase enzyme simply transfers ATP, which is non-cyclic, to cyclic AMP. And cyclic AMP is a famous molecule in biochemical pathways. We call it the secondary messenger. The secondary messenger. And secondary messenger here means the deputy or the delegate of the hydrophilic peptide hormone that couldn't enter the cell, that just bound to a receptor, but its delegate or deputy, well trusted, which is called cyclic AMP, will be produced inside the cell and will do all the necessary to achieve the response that is targeted by this peptide hormone. So we call it a secondary messenger just because it will relay the message of the peptide hormone, or it will relay the message of the hydrophilic signaling molecule. The hydrophilic signaling molecule cannot penetrate the cell, cannot pass across the hydrophobic interior of the phospholipid bilayer. So the cyclic AMP will do the necessary and will tell them that this signaling molecule that is outside the cell wants so and so and so, and it starts to implement or achieve the cellular response. Of course, the cellular response, as we said, might be a change in the shape of the cell, a change in the size of the cell, a change in the metabolism of the cell. And how does this usually occur? As we said, through cyclic AMP. But how can cyclic AMP do all this? Meaning that, again, the cellular response, to wrap up all this, the cellular response is a change, a magnified change. This can be a change in what? It can be a change in the size of the cell. It can be a change in the number of cells when the cell divides into daughter cells. It might be a change in the metabolic rate of the cell, in the metabolism of the cell. These are the aspects of the cellular response. But how can the cellular response produce all this? Through cyclic AMP. And how can cyclic AMP do all this? By two broad mechanisms of action. By managing or controlling gene expression. You know that when genes are expressed, this means that they are transcribed into messenger RNA, and then they are translated into a peptide. So if we control gene expression, this means that we are controlling the production of specific proteins in the cell. So this is number one, by controlling gene expression. So by working on the gene itself. But the second mechanism is away from DNA. This is by adding a phosphate group or removing a phosphate group from a substrate. If you add a phosphate group to a substrate, this is what we call phosphorylation. And if you remove a phosphate group, then this is what we call dephosphorylation. And we will see that some enzymes are activated or inactivated by phosphorylation, whereas other enzymes are exactly the opposite. So if enzyme A is activated by phosphorylation, enzyme B might be inactivated by phosphorylation. Now, since the cell surface receptors initiate the process of signal transduction, so we have to study them in more detail. The cell membrane receptors, those receptors that are present on the cell membrane, why are we always specifying and saying cell membrane receptors? Because, or in order to distinguish them from another type of receptors that are not on the cell membrane, but that are inside the cell, in the cytosol of the cell, and these are called intracellular receptors. Intracellular receptors, or cytosolic receptors, because they are present in the jelly-like cytosol around the organelles. The intracellular receptors can bind to signaling molecules just like the cell membrane receptors can bind to signaling molecules. But the majority, we can say that the majority of the receptors are on the cell membrane, and they are basically the same in their overall structure. Of course, there are differences, but for example, what do we mean by they are basically similar in their overall structure? For example, they are mostly protein in nature, and not only this, but as you know, they should be 3D proteins in order to have an active site and be capable of binding with the signaling molecule. And you might be amazed to see that, or to know that 30% of the proteins in our human body are not present in the muscles, but they are present in the receptors. In order for a protein to be a cell surface receptor, it should span the entire thickness of the membrane. And when a protein receptor spans out the entire thickness of the membrane, we call it an integral protein. So what is an integral protein? An integral protein is a protein molecule that spans out the whole thickness of the cell membrane. Is it important for the cell surface receptor to span out the whole thickness of the cell membrane? Yes, of course, because in such a way it will have an intramembrane domain inside the membrane itself, an extracellular domain, as well as a domain that is present inside the cytosol. And therefore, it will form a good connection between the extracellular environment of the cell and the cytosol of the cell. Peptide hormones and peptide signaling molecules, which represent the majority of signaling molecules, are hydrophilic. And therefore, as we said, they will find difficulty diffusing through the membrane. And here we had better say through the hydrophobic interior of the membrane, through the hydrophobic interior of the membrane. And that's why they will bind to the receptor. But to which part of the receptor? To the part of the receptor that is on the cell surface. Notice that the part of the receptor on the cell surface is in direct contact with the extracellular fluid, and that the extracellular fluid is mainly made of water, and therefore the extracellular part of the receptor is hydrophilic and is suitable to match with the hydrophilic or polar signaling molecule. As we said, this binding that we call reception will induce conformational changes. Where? In the receptor. On which part of the receptor? In the cytoplasmic side of the receptor. And therefore, there will be transduction, meaning that this signal or this transduction means that this reception or receptive act between the receptor and the signaling molecule will induce changes inside the cell. So to transduce means to change from a simple binding process to a series of or a cascade of chemical reactions. So the signal is transduced or transmitted from the outside of the cell to the cytosol, where the signaling molecule, which is considered the ligand, as we said before. So ligand is another name for the hydrophilic signaling molecule that stays outside, or you can also say that it is the first messenger molecule, since it induces cyclic AMP, and cyclic AMP that's formed inside the cytosol of the cell is considered a secondary messenger. Again, we remind you that the first messenger or the peptide signaling molecule or the ligand is hydrophilic, and therefore it cannot pass through the hydrophobic interior of the cell. And once inside the cell, once this change occurs in the cell, in the cytosolic part of the receptor, another molecule called the secondary messenger, which is usually cyclic AMP, will carry out the task and will induce a series of molecular biochemical changes in the cell. We have actually different types of cell membrane surface receptors. What are they? On this slide, we can see a few examples of the surface cell receptors. The first famous type of cell membrane receptors are the ion channel receptors. And as you know, these are receptors that are specific to ions, like sodium ions, like sodium ion channels, like potassium ion channels. And we also have the ion-gated channels. What's the difference between an ion channel receptor and an ion-gated channel? An ion-gated channel opens and closes, whereas an ion channel is always open. So it opens and closes. But it opens and closes according to what? According to changes in that ion, whether in changes in voltage that have been created by changes in concentration of that ion. Sometimes it opens and closes only by changing the concentration, but sometimes these changes in concentration of the ion changes between what and what? Between the outside of the cell and the inside of the cell, create a voltage difference or a potential difference. These changes in concentration or in voltage will result in opening and closure of the ion-gated channels. But we also have the G protein coupled receptors, and they are famous and they are widespread in the body. And the G protein coupled receptors are actually protein receptors on the surface of the cell membrane that are connected with a specific protein called G protein under the cell, just attached, of course, to the receptor. So this is the main body of the receptor, and the receptor is attached to a G protein. So that's why we call them G protein coupled receptors, because they are receptors that are coupled with the G proteins. And upon studying the signal transduction pathways in detail, we will see how these G protein coupled receptors can cause changes inside the cell. And then we have a third type of receptors, which are called the protein kinase receptors, like, for example, the tyrosine kinase receptors. And we will see how these receptors are made of two halves and how they change in conformation and why they are important in the cell. The ion channel receptors and the ion-gated channels, we will first start by talking about them, and they are considered allosteric channels. And when we say allosteric, we mean that they can change in shape. And when they change in shape, they either open or close a gate in a membrane, and therefore they might allow or prevent the influx or the outflux of ion. So we had better say the movement of ions. So they open and close, they allow and prevent movement of ions, like what? Like the sodium channels, sodium ion channels, potassium ion channels, calcium ion channels, chloride ion channels. And upon studying the nervous system in detail, you will discover how these ion channels are important in the transfer, in the initiation, and in the transport or transmission of the nerve impulses. So again, they're very important for normal cell function. And so they are important in initiation of a nerve impulse, and then transmission of that nerve impulse across the axon of the neuron, and then across the synapse for adequate nerve function. But you will also see that the ion channels are important for the resting membrane potential, as we might explain later on in the curriculum. Now, the G protein coupled receptors. We have talked briefly about the ion channel receptors and the ion-gated channels. We will be discussing this more and more when reaching the part on transport. But here we are going to discuss the G protein coupled receptors, or the GPCRs. So as we said before, they span the entire cell membrane. So when you have a cell membrane receptor, if this is the thickness of the cell membrane, so it will span the whole thickness of the cell membrane. So when the ligand or the first messenger binds, so this would be called the signaling molecule, or the ligand, or the first or primary messenger. When the ligand or first messenger binds to this part of the receptor that lies outside the cell membrane, how would we call this part of the receptor that lies outside the cell membrane? We would call it the extracellular domain of the receptor. So this binding between the ligand and the extracellular part of the receptor will induce changes in the conformation or the shape of the cytoplasmic side of the receptor. So this is the cytoplasmic side of the receptor. So we have binding with the extracellular part resulting in changes in conformation in the cytoplasmic part. And this change in the conformation of the cytoplasmic side will in turn activate a G protein. And we will see why the G protein is considered an on-off switch in the biochemical or for the biochemical reactions that will occur inside the cell. So when we say that it is an on-off molecular switch, molecular here means that it can switch on or switch off the chemical reactions or the biochemical reactions, since all biochemical reactions occur at the level of atoms and molecules. So we have two possibilities. Either the G protein is on or off. So when it undergoes conformational changes, it will be on. And it will bind to, when it is on, it will bind to a molecule called GTP, which is very similar to ATP in the sense that it stores energy. It is a high energy molecule. So it is very similar to ATP. And when GTP is activated, or when the activated G protein binds to this GTP molecule, the GTP molecule releases energy, and that energy activates the enzyme adenylate cyclase. And adenylate cyclase, of course, as we said before, will produce cyclic AMP, and cyclic AMP will initiate or activate a series of cellular reactions. And in this diagram, we can see how the G protein coupled receptors that only receive peptide hormones. By the way, as we said before, since the signaling molecule cannot enter the cell, so this signaling molecule is most probably a hydrophilic molecule, meaning that it cannot go through the it is a hydrophilic molecule, and it cannot pass easily through the hydrophobic interior. So here you can see how a peptide hormone will bind with the G protein coupled receptor. This is the receptor and this is the G protein. And as you can see, they are coupled to each other. So this is why we call them G protein coupled receptors, or receptors coupled to G proteins. You can also see that this receptor is a transmembrane receptor. So the G protein coupled receptors are transmembrane receptors because they span out the whole thickness of the membrane. And you can see that we have an extracellular part and then a transcellular or transmembrane part or intramembrane part, and then we have an intracellular part. So extracellular, intracellular, and in between them we have the transmembrane part. When binding occurs, the G protein is activated, as you see here. And when the G protein is activated, it moves towards the adenylate cyclase enzyme, which is also a membrane enzyme. So this is interesting to know that the adenylate cyclase enzyme is a membrane enzyme. So if you're ever asked about examples of membrane enzymes, you have the adenylate cyclase enzyme. And in such a case, it will convert ATP into cyclic AMP. How this occurs in detail, here we have a specific example. We have epinephrine, which is adrenaline. And adrenaline is a peptide hormone that is secreted by the medulla or the center of the adrenal gland or the suprarenal gland, which is a gland that lies just above the kidney, as we said in the previous videos. So we have, this is the suprarenal gland itself. The outermost part of the suprarenal gland is the cortex, which secretes steroid hormones, but the inner medulla of the suprarenal gland secretes adrenaline or epinephrine. Epinephrine is a hydrophilic peptide hormone. It binds with the G protein coupled receptor. We call this the stage of reception, and then comes the stage of transduction. So the G protein is activated, and the G protein separates from, as you can see here, when the G protein is activated, it would separate from the receptor and bind with GTP. The binding with GTP will induce changes in the adenylate cyclase enzyme that would get again activated and convert ATP into cyclic AMP. Cyclic AMP is considered a second messenger, and it achieves its actions, or it actually achieves the actions of epinephrine. That's why we say that the cyclic AMP relays the message. Relays the message of whom? Relays the message of epinephrine that cannot enter the cell. So cyclic AMP will activate a protein kinase enzyme or deactivate according to what is necessary. So, cyclic AMP would activate the protein kinase enzyme, and the protein kinase enzyme will either inhibit glycogen synthesis and promote glycogen breakdown. And actually, these are not two contradictory actions, because where can this occur? This can occur in liver cells. So you know that in liver cells, we have conversion of the extra glucose into glycogen. And this occurs under the effect of insulin. But adrenaline or epinephrine is an anti-insulin hormone. So adrenaline will prevent this conversion. This conversion is called glycogen synthesis, making glycogen. Adrenaline will prevent it. Why does adrenaline prevent it? Because it wants to do the reverse. It wants to break the glycogen stores in the liver into glycogen. So it prevents glycogen synthesis and stimulates glycogen breakdown. And this happens during fasting, when our stores of glucose, when our levels of glucose in the blood are depleted, so glycogen stores in the liver replenish the blood with glucose. Synthesizing glycogen is called glycogenesis, whereas breaking down glycogen that is stimulated by epinephrine is called glycogenolysis.